This is going to be your one-stop shop for cell cycle material. So we're going to go over the interphase, DNA replication, and also cell division here. Um, so there's going to be a lot of stuff you might not understand including cell cascades, but I'll do my best to explain them. So let's get started. So first we're going to start with the interphase, which is the longest portion of the cell cycle. If we go back, it is everything from the beginning of what is called the G1 stage all the way to the G2 stage. In some definitions, it also includes the G0 phase, but for our purposes, I will consider that outside the cell cycle. So in the G1 phase, the cell grows inside to accommodate for cell division. Obviously, you're gonna make more organelles, or you're gonna expand on existing ones. So mitochondria are gonna replicate as well, because you're going to cut in half essentially. So you need enough for 2 cells. So you're gonna synthesize proteins and mRNA you need for DNA synthesis and replication, because that's going to occur in the S phase. So it prevents cyclin-dependent kinase activities in 3 different ways. So let's talk a bit about what kinases are. Kinases are enzymes that phosphorylate or dephosphorylate certain molecules, specifically proteins, to activate or deactivate their functions. And it does this because when you phosphorylate a molecule, it changes its conformation, so then it can fit into certain receptors. It can be better recognized, or it fits to another enzyme. You can open up the active site or increase the binding rate. So all that kind of good stuff. So PRB binding to E2F to downregulate S-phase cyclins. So essentially what is happening here is PRB is a protein binding to another protein called E2F, and we're going to deregulate cyclins to downregulate S-phase cyclins. So the anaphase promoting complex activates, so it's going to create S and M cyclins. The reason we want to do this is we don't want to begin replicating our DNA too early, and we don't want to begin mitosis too early. And CDK inhibitors are in high concentration because we want to stop the CDKs, cyclin-dependent kinases, from activating. So cyclins are what are going to activate certain kinases to function. So we're trying to get rid of these cyclins and specific cyclins as well. So ATM and ATR, which are 2 protein complexes, check for DNA damage before we move to S phase. So we want to make sure our DNA is okay. There's no problem with our genetics. There's no significant mutations before we go on into the DNA synthesis. Otherwise, that error is going to be synthesized into our new cells, and it can actually then propagate forward. So this is sometimes what happens in cancer, is that the DNA damage is not detected, and tumor suppressing genes are then demolished in the DNA or damaged in the DNA, and so the cells then become a tumor because they have unregulated division. So the p53 tumor suppressing protein and G1S cyclin formation are all part of that checkpoint. So p53 tumor suppressing protein comes up a lot, and it's a protein that can cause apoptosis, which is cell death, programmed cell death. The S phase is a DNA synthesis phase. So here we're going to replicate our DNA. So the SYN3 or CIN3 CDK2 complex promotes transcription of S phase genes. So we're still making proteins for S phase to get ready. And the complex suppresses repressor WE5 or WH5. Histones are synthesized, and what histones are, they are protein complexes that DNA can wind around to condense. So this actually helps us fit our DNA because DNA is quite a large molecule. And on different chromosomes or on different portions of the DNA, This is actually how we can keep some genes repressed or out of use, some introns, specifically some portions of our DNA, because they'll be so tightly wound on the histone they can't be used, and some of the looser parts can then be transcribed. Histones also help bind certain repressors. So the replication checkpoint detects a stalled replication fork, and which we're going to talk about in a second, is how DNA replicates. But they create something called a replication fork. We want to make sure our DNA is completely replicated before going forward. So we check for this stalled replication fork to make sure our synthesis is actually going through all the way. The SM checkpoint blocks mitosis till replication is complete. So this is the DNA synthesis to mitosis checkpoint, blocks mitosis till replication is complete by inhibiting cyclin B CDK1 complex. CDK1 is very important for mitosis. Or meiosis. This is actually what's going to start the process. CDK1 cyclin B complex this is the complex that says, okay, let us get going with mitosis. And again, we use ATR and ATM to then repair and detect damages in DNA during our synthesis. In case we put in a wrong nucleotide, we match A with a G instead of with a T, or we match this, or whatever the nature it is. So let's actually talk about DNA's replication, what's happening in that S phase. It is a semi-conservative process, as I talked about in my experiments to know video. It was proven by the Mendelssohn-Stahl experiment. One strand is kept completely intact, and a new strand is synthesized. And since DNA is a double helix, there's 2 strands. So from 2, we make 4 strands total and 2 new pieces of DNA, with the original strand having 1 portion of the helix in each new DNA. So if we had a DNA like this, we go through replication, we have a DNA like this. And notice how the original 2 strands are now with a new strand. And then it can propagate to 8, 16, blah, 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 as we get more and more into cell division. SCDK2 and CDC7 activation facilitates the assembly of the pre-initiation complex that binds to the origin, ORI. ORI is a big buzzword for DNA replication or any kind of synthesis for genetic material, because this is where the DNA replications begin. This is going to happen in multiple places. Again, as I stated, DNA is a super long molecule. You're not going to do just replication starting at one end and go all the way down to the other. No, you're going to have multiple origins of replication, and you're going to replicate to make the process faster, and you're going to have multiple proteins doing that. M&M helicase or DNA helicase is in this pre-initiation complex and is activated, and it unwinds the DNA helicase. So it from a double helix, we become kind of like a ladder, and it's going to start breaking those hydrogen bonds between the nucleotides to then allow for the synthesis to go through. It creates a replication fork, because we don't unwind all the things. So you can't, I have a picture somewhere. Here. This is kind of what a replication fork looks like. Right here, it's kind of, this is considered what the replication fork is. I also, at least if you're getting confused on how to look at this or what this might look like or what I'm saying, I highly recommend watching an animation on this to understand how things are occurring. So DNA polymerase is actually what's going to add nucleotides to create our new strands. It synthesizes in a 5' to 3' direction, creating both a leading and a lagging strand. So 5' and 3' refer to where depending on what the sugar phosphate backbone looks like. So one strand is 3' to 5', the other is 5' to 3'. But because we are synthesizing in a 5' to 3' direction, and one of the strands is going backwards, so that creates a lagging strand. But in the lagging strand has what we call Okazaki fragments. So there's an RNA primer that's added to the lagging strand, so then it can be added, in the correct direction, but you have to keep doing this. And what it creates is called Okazaki fragments, as they're small portions of DNA, RNA that then need to be completely zipped together by a protein that's going to come in. RNase removes the RNA from the primers, from the DNA. So we don't have a mixture of DNA, RNA. We have pure DNA. And again, the difference between DNA and RNA is the deoxyribose versus ribose. So deoxy means there's not the oxygen molecule in the sugar, and ribose does have that sugar. So different polymerases have different functions. So there's actually multiple polymerases. So in E. coli, I believe it's polymerase 3. It is polymerase 3. Is responsible for the main synthesis, while polymerase 1 is what is replacing RNA with DNA. So, it's not just one super protein doing all this work. There's different types of polymerase that that is classed together as a polymerase because they do similar functions and they work on the same job. This is kind of how there's multiple kinases. There's multiple polymerases as well. The replication complex utilizes a sliding clamp protein to prevent dissociation. So you don't want your protein to go away from your DNA before you're done replicating. So there's a sliding clamp protein to keep it in place, and it slides along the DNA. So it clamps onto the backbone and slides along with the complex to synthesize the DNA. There's a 911 complex that stops/inhibits DNA replication. So this is where we're going to say, okay, we're done with this portion of DNA. You can disassociate now. So as you can imagine, when you unwind something, you build up a lot of tension at the other ends, especially if you are doing it just in the middle. So if you need an example of this, grab 2 pieces of string or rope, wind them together, pull the middle apart, you'll see the tension go to the ends. This is very bad for DNA because that could cause a lot of damage. So what our bodies have done and organisms have done is they have topoisomerases. And topoisomerases are proteins that prevent that tension and that loosen that tension to allow replication to go smoothly without damaging the rest of the DNA. And this allows us to have multiple origins of replication as well. Otherwise, we would just need just one with topoisomerases going all the way down. And again, I would watch animations to better understand the process of it. It is a very visual and a very active thing. This is a simple polymerase, or simple example. This is polymerase alpha, which I believe is the human polymerase. And it is going to perfectly synthesize the DNA in the leading strand going all the way up, because this is 5' to 3'. But in a lagging strand, such as this one, we have Okazaki fragments, such as this, which have a DNA or RNA primer and the DNA attached. And then we have just some missing portions, which then a different polymerase will come back, remove the RNA, replace the RNA with alongside with RNase, and then add the correct nucleotides. These are binding proteins that also help. These are not the sliding clamp ones, but these also help keep the complex onto the DNA. This is the topoisomerase, which is helping protect the tension go through entire DNA and damage it. It's a lot of information, so take your time digesting it, watch a lot of information about it, watch a lot of different animations about it, keep re-listening, keep re-watching, because this comes up a lot. It's very important that you understand this. It's one of the more fundamental questions for biological scholastic bowl. They might ask about polymerase, they might ask about Okazaki fragments, they might ask about helicase, they even ask about topoisomerase. It could be a clue for RNA. It's a question for kinases as well. E. coli, it shows up as a model organism question. Obviously, the Mendelssohn-Stahl experiment. So this will get you quite a few toss-ups, quite a few bonuses, a lot of points in general by understanding this process really well. So again, I suggest taking your time and understanding it. And again, please feel free to ask me any questions about it, and I'll do my best to help you. Okay, after we finish DNA synthesis, we go through the G2 phase. So there's some stuff we just still need to do before mitosis. So it's a period of rapid cell growth and protein synthesis. So we're going to synthesize proteins we need for mitosis. We're going to synthesize proteins for just our general function as well. It's important to note that during the cell cycle, cells still have a job, and they are performing that job. It is not technically a necessary phase. There are a lot of organisms who skip the G2 phase. However, a majority of organisms do the G2 phase, and it's thought to be for cell growth regulation, again, to help detect for tumors, to help prevent tumors from occurring, and from cancer cells propagating themselves too quickly. When it ends and mitosis begins when the threshold for cyclin B1 CDK1 complex is met. So this is one of the things we are synthesizing. This complex is also known as the maturation promoting factor. This is what triggers centrosome separation, nuclear envelope breakdown, and spindle assembly. Some of you who have studied biology before might recognize that is the beginnings of prophase. And the WEE1 or WEE1 inhibits the complex formation. So we do need to also denature WEE1 as we are going through. WEE1 is part of the checkpoint of the G2 cycle. This checkpoint involves DNA for damage again. We are always just checking our DNA for damage. That's one of the takeaways from this. And it also promotes CDK1 activity, allowing cyclins to bind to it. And if you haven't caught on, CDK stands for cyclin-dependent kinase. So certain cyclins have to bind to different kinases to then activate that. Okay, so this is the G0 phase, as I mentioned. So this is the cellular phase outside of the cell cycle. It can be reversible or irreversible, so quiescent or senescent. You may recognize these as terms from stem cells. G0 phases are often stem cells. The sentient state occurs when DNA damage is too significant. This is a cell that's probably going to undergo apoptosis when everything is all said and done. It could also be specialized cells like neurons. Neurons don't generally replicate. So the G0 phase is the phase they are in. And stem cells are quiescent G0, reversible G0 phase cells. Either phase that enters G0, the body says, hey, we need some of these cells through signaling hormones or signaling proteins. And then the cell says, okay, if it's in that location, and then begins doing that. RIM15 or nutrition signaling leads to a cell to G0 entry. RIM15 was first found to play a critical role in initiating meiosis in diploid yeast. So this is a good clue for diploid yeast or yeast in general. Something else I should mention is if you hear me say things like RIM15 or WEE1 and you're just curious, like what the hell is that? Those are proteins or receptors depending on their function. I wouldn't worry too much about what they are rather than knowing when they are present, what they do, and knowing they are buzzwords for certain things. Okay, so that is the end of the interphase, and now we're going into cell division. There are two forms of cell division we are going to talk about, mitosis and meiosis. I just do want to make a quick mention that I do not separate the prophase, metaphase, anaphase, telophase, cytokinesis into their general, I don't split them between mitosis and meiosis, what I'm trying to say. Generally, it's galactical. They ask them as they are just one and the same. Now, sometimes they will say it's meiosis dependent or mitosis dependent ones at the end of it. Sometimes they'll give clues that are specific to meiosis 2 or meiosis 1. or I'm sorry, like metaphase 1 or metaphase 2. But you can just say metaphase and they will give you the point. So I just want to make that note. Again, this is not meant to be a test review. This is meant to get you through scholastic world questions. I just want to make that differentiation clear. So this is mitosis, a lot going on. Let me break it down. Mitosis is basically cell division forming two equal daughter diploid cells. It is heavily regulated throughout the checkpoints, but still sometimes something happens, and that's when cancer cells replicate without regulation. MAP kinases are named for being part of mitosis. The M is for mitosis. RAF or RAF, they sometimes say, kinase activates lower accessory kinases in a cascade, like ERKs. So one of the things I want to explain, takes a moment to explain, is a cascade. A cascade is the idea of we have a parent function that activates 2 more functions, that activates 4 more, that activates 8 more. It's an exponential growth. So RAF kinase will activate another 3 kinases, let's say. It manages to get 3 kinases. One RNA kinase activates 3 kinases. And that each one of those kinases is able to activate another 5. Oh, then we're hitting 15. And then each one of those is then able to hit another 10. I'm activating 150 ERKs. The reason this is so important is because as small as you think cells are, these molecules are smaller and fit inside the cells. So the cascade helps move things along faster, signals the cell, go, 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 go. We are doing this, we need to do this now. There's a checkpoint actually in the metaphase to see if the bipolar spindle connection to the chromosome. So here, this is a spindle, and this is a chromosome. We just want to make sure that these are actually connected, so we're actually separating them. So we're not creating any genetic defects in either of our cells that we then have to do apoptosis. So Thomas Hunt actually studied the protein concentrations during mitosis in sea urchins, and he noticed the concentration of different proteins being higher at certain points and lower at other points, which makes sense because during mitosis we need different proteins. So this is a count of chromosomes and chromatids. Chromosomes are the X's, chromatids are each of these smaller ones. Yeah, and this is just the number of how many are present in each portion. The reason the number of chromosomes double after metaphase is because we separate, so chromatids and chromosomes become the same thing. But each cell only ends up with 46 chromosomes at the end of it. If you're wondering why it's 40, there's 23 pairs of chromosomes, so 46 chromosomes in total. Okay, so meiosis is we're creating 4 unique daughter haploid cells, so gametes, germ cells. This is our sperm or eggs. So aneuploidy, which is an abnormal number of chromosomes. So this is something like trisomy 21, also known as Down syndrome. It's going to occur in meiosis. Down syndrome specifically is an example of a non-disjunction, which means that we have an extra chromosome. An extra chromosome was pulled over in the creation of the germ cell. And one of the big clues for meiosis is that tetrads will form. So this is a tetrad where 2 pairs of a father and a mother chromosome line up together. This is where it's going to form. It's going to establish a chiasma. And this is a complex where crossing over can occur. So this is where genetic information is going to swap. And this is how we create unique individuals throughout reproduction. And these are all the different recombinants. This complex is known as the synaptonymal complex, or synaptonymal complex. They might say it wrong. So I would know these are good buzzwords for meiosis. And worst come worst, they'll prompt you, and you will say metaphase. So leptotene, pachytene, and zygotene are actually unique parts of prophase 1 in meiosis. So meiosis in our prophase and meiosis and metaphase are different because they need different preparations. Because meiosis has to undergo 2 divisions for haploid cells. Mitosis is just a singular for a single cell. So that's why there is these unique portions here. Just knowing these names is sufficient to know that they are talking about meiosis. Or the answer might be prophase, but it's hardly ever prophase. It's usually meiosis. Good tip, actually, I should mention now. Generally, they only ask about mitosis, meiosis, anaphase, metaphase. Those are like the 4 big ones if they are talking about cell division itself. They can ask for questions within cell division, like they can talk about kinases and things such as we've talked about before. Cascades sometimes shows up, actually. So I would just note that those are the 4 big ones that come up, but prophase still does show up once in a while. Okay, so what's actually happening in the prophase? And again, this is a combination of mitosis and meiosis ones. Chromosomes condense and centrosomes begin to move. So this is essentially, we're saying, okay, we are getting ready to split these chromosomes apart. Condensin, which is a large protein complex of 5 subunits. Each subunit can be different in different organisms to cater to specific needs of those cells, is utilized to condense the chromosome. The centrosomes are going to be moving to opposite ends of the poles. These are powered by associated motor proteins, so these are proteins that actually facilitate movement. These interpolar microtubules are going to help position the centrosomes correctly. So these microtubules are going to be able to detect based on the location, oh, I'm supposed to be here. This is where the pull of the end, so we can pull them apart, far enough apart. That's kind of a simplistic explanation of it, but it's the explanation that works the best, I would say. In plant cells, microtubules gather at opposite poles and spindle apparatuses form at foci. Also, something called a phragmosome might be required. How do you explain a phragmosome? I guess the best way would be just to simply show you what a phragmosome is. Hopefully this shows up in the video. Otherwise, just Google it, and it's the Wikipedia page I'm looking at. So a phragmosome is simply a sheet of cytoplasm that is needed for plant cells because of the existence of a vacuole, usually taking up majority of the space. I just know that a phragmosome is a term generally used for plant cells. It says this happens in D2, but it's actually utilized in mitosis. So how it are in prophase and cell division. So I would answer more along prophase. Answer could be cytoplasm, plant cell, or in preparation for mitosis. They don't do this in meiosis because of the beginning cells are inherently different in mitosis and meiosis. So, your potential answers if you're a fragmosome is plant cell, mitosis, or cytoplasm. And you should be able to answer that simply based on what they're asking for. They're asking for this process. Oh, mitosis. Oh, this substance, cytoplasm, or these cells. Oh, plant cells. So, you use your intuition there. And as you play more and more, you'll get better at listening to what clues or what they're actually asking for. The nucleoli will break down, stopping ribosome production. So this is where we're going to say, okay, we're going to stop synthesizing proteins actually. So proteins can still degrade, which is what I was talking about with Thomas Hunt, to prevent, repeats or whatever is going to happen. So things can degrade. We can actually still make proteins with the existing ones, but it slows down significantly. The nuclear envelope is still intact here. And in meiosis, this is where crossing over occurs. I'm going to be honest, sometimes they say it occurs in metaphase and scholastic bowl for whatever reason, but the answer should be prophase. So just be careful. If they start talking about crossing over, I would go ahead and just say meiosis, and they'll prompt you if they need to. Metaphase, microtubules connect to the connector cores, and chromosomes line up on the namesake plate, which is the metaphase plate here. So this is like exactly at the center of the cell. A cell cycle checkpoint during this phase is to ensure that they are properly lined up and that these spindles are actually connected. This is also sometimes referred to as the spindle assembly checkpoint. Secure destruction by CDC20 allows for the transition into anaphase. So this ends the checkpoint. And P31 is activated by MAD1, MAD2 complex, leading to release of MAD2 from CDC20. And metaphase consists of 4% of the cell cycle. A lot of stuff here. Be careful if you're going to buzz in with securin. This does show up for anaphase questions as well. I would say the safer bet is hearing MAD1 or MAD2. Also saying, oh, securin ending or beginning this process. That's actually very important to hear. Otherwise, you're taking a 50-50 shot stab in the dark. So be careful if you're going to buzz in on securin. Nothing else I have to mention here. Okay, anaphase. This is actually where we're going to see the chromosomes split into chromatids or into their haploid variants. So what's happening is these microtubules are actually depolymerized. So they are polymers, and they actually depolymerize at a rate of 50 subunits per second to shorten them, and then to actually separate the chromosomes, because there is some force required there, because they are connected through bonds. Motor proteins help rip them apart. The motor proteins are dyneins or kinesins. The later portion of the stage sees the interpolar microtubules at each centrosome push apart. Astral microtubules connect centrosome and join the cell membrane. So now we're getting ready to start, orienting ourselves. Okay, we need to start splitting things in half and getting ready to cut our cell in half. Anaphase begins with ubiquination of, this should say securin, by the anaphase promoting complex. Remember that from all the way from the G1 phase, it's back now. And another clue is this degradation of cyclin B is necessary to start the anaphase. Telophase, this almost never shows up as an answer, but I figured if it does, you better know it. The nuclear envelope is reassembled, the nuclei reforms, and chromosomes decondense. This is kind of the anti-prophase. The mitotic spindle is disassembled, and microtubules are depolymerized again, even further, to ensure that they are not taking up space in the cell. It's mainly driven by the dephosphorylation of CDK substrates. All of these we've talked about, the APC, anaphase permanent complex, CDC20, etc. They're all dephosphorylated. So generally we think of phosphorylative as active, dephosphorylative as inactive, is how you can simplistically view this. There are some experimental studies that show that chromosome distance may trigger spatial clues for telophase. So through the anaphase, when they're getting pulled apart, how far the chromosomes are pulled apart can actually begin the start of telophase. Again, this is not set in stone yet. This is conjecture mainly, but it could show up. CDC48 is an important regulator of 48. This is actually what employs ATPase and other kinases and other enzymes to actually do it, what I described in the telophase. So disassembling the spindle, assembling the nuclear envelope, decondensing the chromosomes, all of that jazz is done because of CD40 being an important regulator. Okay, cytokinesis actually does show up once in a while. So it's the cytoplasm of the single cell dividing into two cells. When we say cytoplasm, we just don't mean cytoplasm. We also mean the organelles. This is what is called as the cleavage furrow forming and allowing for the equal separation of material. This is an actin-myosin complex is the protein duo responsible for here, for pinching the cell apart. Myosin is phosphorylated in the light change to help activate actin. And the cell wall in plant cells through the cell plate are formed to allow for the separation of the two cells, because you need a new cell wall for your new side, right? Because you have a rectangle. If you just split it in half like this, everything's going to fall out. So you need to actually add a wall in the middle here to allow it to separate equally. In oogenesis, this is a specific form of meiosis. One cell gets a lot more material, and that's the one that becomes the egg. So in meiosis, for formation of egg cells, which is called oogenesis, we actually only form two viable cells, and the other two are discarded generally. Cellulose is secreted after pectin to create the middle lamella. This is a portion of the prior to the cell wall, prior to the cleavage furrow. This is like the middle lamella is going to define the center kind of thing. In algae, pit connections and pit plugs are formed during cytokinesis. This shows up once maybe. But this is a good clue for cellulose to know. What am I saying? Middle lamella only shows up for the cell plate. It is not in animal cells. I should not say it occurs in the cleavage furrow because it's cellulose. My apologies. Okay, this is the last thing we're going to talk about. It's apoptosis. This is what we call programmed cell death. It is commonly caused by p53, a tumor suppressing protein, detecting damaged DNA from the earlier complexes. And it causes the release of cytochrome C from the mitochondria. So when cytochrome C is released, it's released by the BAC and BAX proteins. And cytochrome C acts as a signaling molecule. There's a bunch of cytochrome C. There's not just one or two in the mitochondria. There's a lot. And there's also quite a few mitochondria. So this is a massive signal to say, hey, we need to start our cell death, our programmed cell death. This is performed through blubbing of the plasma membrane. This is an example of what blubbing is. You can also see it here. It's like molecules condense, or the cell membrane condenses into itself and starts peeling away pieces. And then phagosomes can come eat them and safely remove the material from the bloodstream or whatever portion of the body they are in. Ligands binding to the FAS or FAS receptor triggers the apoptosis. So cytochrome C would bind to this FAS receptor, and that's what's going to actually trigger. Or I can't remember if it's p53 or cytochrome C, but FAS receptor is a good buzzword for apoptosis. And what ligands are, if I never explained that, they are things that either get chelated to metals, or they are, in biological and biochemical terms, these are molecules that bind to receptors. This was studied heavily in C. elegans, which is one of your model organisms. And one gene specifically that was studied heavily was the SED4 gene. And Diablo binds to the XIAP inhibitor to allow this process to go through. So to allow for apoptosis, Diablo needs to bind to this inhibitor to prevent the inhibition. And that is everything for your cell cycle stuff. This will get you most of the way through, get you a lot of good questions. And again, I don't think I iterated this in my experiments video, but I want to iterate this one. You don't need to know everything in these PowerPoints. Mainly what I talk about first, or what's at the top, is more important to know. Things that could show up in bonuses. If you know everything in this PowerPoint, you are going to be a very good player for answering cell cycle questions. So, as you progress along, JV, we would expect them to know the first. We would expect them to know less than we do our varsity. But if you want to be a very good player, a star player, knowing at least 70% of what's in all of these PowerPoints will get you there. So I just want to iterate that, and so don't feel like compelled to know everything that's in these videos, but feel compelled to know a lot of it. So, but that's the end of it. So thank you for watching. Again, send me any feedback. Send me any questions you have to the email in the description. And you're all set.